to the 6th Annual International Remote Conference Science and Society 2021. My name is Dr. Ishita Gupta and I'm an MBBS graduate from India. I will be presenting my research on hantavirus, a conspectus of a rodent-borne infection. My co-authors are Dr. Ritu Teshkara and Dr. Amit Barua. These are our disclosures. We have nothing to disclose, neither do we have any conflicts of interest. Background. In the fall of 1992, there was an exceptional amount of rainfall in the four corner regions of the United States. This led to the growth of a massive deer mouse population during the following year. The causative agent of this disease, a hantavirus identified by the CDC by means of serological testing, was designated as sin nombre, meaning no name virus. The syndrome associated with it became known as the hantavirus pulmonary syndrome. About 200,000 cases of hantavirus infection are thought to occur annually worldwide. The case fatality range from 5 to 15% for HFRS and up to 40% for HPS. As of January 2017, there were 728 cases of a hantavirus disease reported in the United States. All laboratory-confirmed cases of hantavirus pulmonary syndrome, and cases of non-pulmonary infection have been included in this. Hantavirus has been shown to infect various types of cells in the human body by its ability to attach to the host cell surface receptor by using glycoproteins present on its surface. Some commonly affected cells are macrophages, lymphocytes, dendritic cells, endothelial cells, follicular cells, and epithelial cells. Hantavirus infection shares signs and symptoms with leptospirosis, thus risking misdiagnosis. This virus has shown to cause three main syndromes. Management of the infection primarily focuses on supportive care as there is no specific treatment for the infections up to date. Our objective is to review the literature on hantavirus. We focus on the structure, pathogenesis, clinical features, suggested treatment, and prevention. We also lay emphasis on the patterns of virus outbreaks and the abnormal transmissions that have been recorded with this virus. Our methodology. We conducted an electronic search between April 2020 to May 2020 for articles on hantavirus using two primary databases, PubMed and Google Scholar. The mesh keywords used were hantavirus disease, hantavirus outbreak, hantapulmonary syndrome, hemorrhagic fever, and was comprehensive with cross-checking of reference lists from the articles retrieved. Full-text peer-reviewed articles published in English and research articles on hantavirus were included in our review. We had 70 articles for our final review, and each paper was reviewed by two authors independently. Disagreements were discussed amongst all reviewers and resolved via consensus. We tabulated the data using Excel, and referencing was done using EndNote. This study did not require ethical approval as data was obtained from already available databases and direct patient or animal involvement was not present. Results. Annually, around 200,000 cases of hantavirus infections are reported worldwide. 60,000 to 100,000 cases of HFRS cases are recorded worldwide. HFRS is mainly caused by the pumila-virus, hantavirus, and CO virus. Geographically, hantaviruses can lead to hemorrhagic fever with renal syndrome in Asia and Europe, and hantavirus pulmonary syndrome in North and South America. Evolution of hantavirus. Pleiseman et al. In 1995 defined the genetic variation at the nucleotide level. They found it to be 0.2 to 1.2% within individual rodents, 1 to 2% within a population, and up to 17% between lineages. Ramsden et al. Suggested that hantaviruses exhibit short-term evolutionary rates equivalent to those seen for rapidly evolving RNA viruses. A long-standing coevolutionary history of hantaviruses and their rodent carriers have been suggested by phylogenetic analysis. Congruence can also develop when host switching occurs, followed by pathogen speciation. Species jumps have occurred during the evolution of insectivore-borne hantaviruses, and also also between Microtus viale and Khabarovsk virus and Lemons-carried Topographov virus. Ramsden et al. Believes that our ability to predict the scale of future host germs is affected by the various gaps in our understanding of the evolution of the hantavirus. Reassortment. Reassortment is the exchange of genetic segments leading to genetic diversity. Being a segmented RNA virus, hantavirus is capable of segment reassortment, which is one of the ways viruses can achieve increased virulence, leading to increased infectivity and adaptability to new hosts. Thus far, although natural reassortment between different strains of similar serotypes within populations of the same rodent species have been reported. In addition, reassortment between genetically distant related Synombre virus and Black Creek Canal virus have also been reported in nature. Reassortment in genetically unrelated hantavirus by simultaneously infecting Vero E6 cells with Aedes virus and Synombre virus has been demonstrated, giving rise to the possibility of generating new virus genotypes with increased virulence. The reservoirs of hantavirus are rodents, like the bank voles and the yellow-necked mouse. Insectivores and bats serve as hosts for the hantavirus. Mode of transmission is by the urine and the feces of rodents. The rodents can also excrete the hantaviruses in the saliva. Human infection is mostly via inhalation of aerosolized virus-contaminated rodent excreta and contaminated food. However, 2 reports of percutaneous transmission and a suspected case of perinatal transmission have been documented. Ecology. Rodents were considered as primary hosts for hantaviruses. As a consequence of the geographic distribution and natural history of primary rodent hosts, hantaviruses have been able to cause outbreaks across the globe. However, later, bats were added to the list of reservoirs. Hantavirus detected in larger animals like red fox, domestic cat, and dog have negligible to no risk of human infection. The infection of these animals is considered to be a spillover infection. Hantaviruses closely resemble its host in terms of ecology and geographical distribution. The temperate climate of Central Europe supports the increase in rodent population. The warm summers that result in mass years, that is where abundant nuts of forest trees fall into the ground and provide nutrients to rodents, has shown to contribute to the increasing rodent population. In the non-mass years, less variation is noted in the rodent populations, which may not guarantee efficient transmission of hantaviruses. Freezing temperatures and a sudden warm spell that melts the snow has led the bank voles to migrate closer to human dwellings. A few studies from China proved that a good harvest, low rainfall, and flooding may vouch for breeding of field mice. Studies from North America suggested that precipitation may aid in the increase of rodent population. Urbanization, colonization, deforestation, and agricultural development may lead to increased risk of vector-borne disease, as it favors the interaction between humans and the possible reservoirs of hantaviruses. Epidemiology. In North America to date, more than 2,000 cases of HCPS have been identified in small clusters. A total of 616 cases occurs in the United States. The first outbreak was in May of 1993 when an unexplained pulmonary illness occurred in the Four Corners of the United States. The second outbreak was witnessed many years later in 2012 in Yosemite National Park. 10 confirmed cases of hantavirus infection were reported, with 3 being fatal. The third and the most recent outbreak was by the Seoul virus in 2017. In Canada, active surveillance for hantavirus pulmonary syndrome began in 1994. It was made a nationally notifiable disease in January 2000. In Canada, the ubiquitous deer mouse is the primary reservoir of Synombre virus. As of the 31st December 2014, a total of 109 cases of hantavirus pulmonary syndrome were recorded in Canada, with an average of 4 to 5 cases per year. Most of the cases of HPS have occurred in the western provinces of Canada, with 1 case reported in eastern Canada. The 5 pediatric cases of hantavirus was described by Lee et al. In 1997 in Canada. 2 imported cases of hantavirus pulmonary syndrome have also been reported in Canada, one from Bolivia and one from Argentina. Hantavirus infection in rodents and humans in South America has been recognized since 1982. In Argentina, there are about 1,300 HCPS cases identified till 2013. An outbreak of 20 HPS cases in the southern region was reported in the year 1996, which for the first time provided strong indication of person-to-person transmission, as each affected person had direct contact with the previously affected patient. An abnormal outbreak was detected in Argentina in 1995, when thrombocytopenia was detected in a mother who died due to respiratory failure 8 days following childbirth. Her child was diagnosed with Andes virus after developing respiratory symptoms 4 days later. This case gave rise to the possibility of considering new routes of transmission, that is intrauterine, perinatal, and/or breastfeeding. In Europe, the Pulmonary hosted by bank wall that inhabits throughout Europe is a commonly found strain of hantavirus. The Pulmonary causes a milder form of the disease, Nephropathica epidemica. The virus infections have been reported from Finland, Germany, Belgium, Russia, Croatia, France, Sweden, Denmark, and the Balkan countries. In 2018, 29 countries in Europe reported 1,826 cases of the hantavirus infection. From 2014 to 2018, the infection rate fluctuated between 0.4 and 0.8 cases per 100,000 population. Russia. From 2000 through 2017, 131,590 cases of hemorrhagic fever with renal syndrome have been reported, which were caused by 6 different hantaviruses. In China, accounts for 90% of the global HFRS cases, recording 20,000 to 50,000 cases annually. South Korea is another country which is endemic for HTNV. Cases have been reported in Thailand, Indonesia, and Taiwan. In South Asia, although hantavirus infections have been reported in India and Sri Lanka, the exact species have not been studied. However, no outbreaks have been reported till date. In Africa, antibodies against hantavirus have been detected in the population, but no study regarding the prevalence of the disease is yet available. Structure. Hantavirus is a lipid-enveloped RNA virus consisting of 2 surface glycoproteins. The RNA genome comprises 3 segments, short, medium, and long. These are responsible for coating of nucleocapsid protein, the 2 envelope proteins, namely G1 and G2, and a polymerase respectively. Pathogenesis. Hantavirus has been shown to infect various types of cells in the human body. The virus has been proven to use entry ports like integrins, beta-1 and beta-3, calculain-coated pits, and many other proteins in the cells lacking integrins. Many studies have also shown deaccelerating factor, that is DAF, and CD55 as an important cofactor for the infection. Clinical cause and syndromes. Sudden onset of fever, mostly with chills, malaise, and abdominal pain, is suggested to be the hallmark of the disease. 3 main syndromes of hantavirus infections have been noted: hemorrhagic fever with renal syndrome, Nephropathica epidemica, and hantavirus pulmonary syndrome. Laboratory findings. Laboratory findings are consistent with leukocytosis, atypical lymphocytes, thrombocytopenia, and increased serum creatinine. Immunohistochemistry is used to detect hantavirus antigen. Hantavirus IgM titers are detected in the fresh and recent cases, while rising IgG titers are detected in the relatively old and recovering cases. PCR is used to detect viral RNA in the host. Hantavirus antibodies may be detected even years after the infection. Treatment. At present, no specific treatment is available for hantavirus infections. Treatment is primarily supportive. HPS patients must be provided with oxygen, if necessary intubated and mechanically ventilated. They must be regularly monitored for changes in blood pressure and fluid status. Analgesics like paracetamol, antibiotics for secondary bacterial infections may be administered, however, NSAIDs needs to be avoided. A controlled study conducted in China showed that the administration of ribavirin in the early phase of treatment of HFRS can reduce mortality and severity of symptoms. However, definitive evidence of benefits of using ribavirin in HPS has yet to be established. Prevention. The most effective method to prevent hantavirus infection is to avoid human contact with rodents and their excrement. Preventive measures include using rubber gloves and wearing masks or handkerchiefs while cleaning. Keeping trash in closed containers or tanks resistant to rodents. Keeping grains in airtight or sealed containers out of reach of mice. In addition, traps can be set for mice and rats. Grass and weeds must be cut short around homes. Surfaces must be wetted with water or disinfectant before sweeping and cleaning, and waste material accumulation should be avoided. Vaccination is another mode of intervention that is considered to prevent hantavirus infection. To date, several types of vaccines are being researched upon, which include inactivated vaccines, DNA vaccine, particle-like vaccines. However, only inactivated hantavirus vaccine, Andes virus DNA vaccine, and a combination of PUV and HTNV vaccine have reached the clinical trial phase so far. No FDA-approved vaccination is available, though China has approved the use of bivalent hantavirus vaccines based on inactivated HTNV and COV and included in the National Expanded Program on Immunization since 2008 in China. Since then, the cases of HFRS has fallen to less than 20,000 cases per year. Conclusion. Hantavirus disease can present as various clinical syndromes, as the virus has been shown to infect multiple types of cells in the human body. There have been numerous outbreaks of hantavirus throughout the globe. Thus, the fact that hantavirus has the potential to cause a worldwide pandemic cannot be ignored. It is therefore imperative that appropriate measures are adopted to minimize human interaction with rodents. Meanwhile, a potent vaccine against hantavirus must be invented and a specific cure must be discovered. Besides enhancing the host immune responses against this viral infection, appropriate preventive strategies and support and treatment of complications would also be crucial. Thank you. Please feel free to reach out to any of the authors should you have any thoughts, comments, or questions.